Generally, catalysis by biological enzymes can be very useful in the pharmaceutical industry. Biological enzymes are capable of performing many isomer-specific reactions, along with many derivatization reactions reacting specifically at targeted reaction sites. The use of biological catalytic enzymes can do reactions ranging from very specific reductions of ketones by ketone reductases and carbon-carbon double bonds by enodate reductases to very general reactions such as oxidation by oxygenases. Enzyme catalyzed processes are often more efficient than typical synthetic processes. This can aid in more sustainable production of pharmaceuticals by reducing isomeric waste and reducing the requirement for derivatizing agents.
Diabetes is a very common disease affecting 1 in 10 in the US and 1 in 15 in New Zealand. Diabetes occurs when too much glucose is in the blood. This is usually caused by the pancreas not producing enough insulin. A person with diabetes will usually have 0.072 to 1.62 grams per liter of glucose in the blood, whereas someone without will have 0.072 to 1.4 grams per liter, depending on the time of day. Insulin is responsible for two main things. First, it transports the glucose from the blood supply into the fat and muscle cells, where it can then be used for energy, and secondly, to switch off the liver once the level of glucose in the blood is high enough. Therefore, if there is not enough insulin being produced in a person, then the glucose levels remain higher in the blood, causing the well-known disease diabetes. There are multiple types of diabetes: type 1, where the person produces no insulin, and type 2, where the person makes insulin, but they are either resistant or their production is sluggish. Attempts as early as 1889 to find solutions and understand diabetes were made. Oskar Manowski and Joseph von Meng found that when the pancreas gland was removed from dogs, the dogs then developed diabetes. It was later suggested by Albert Sharpe Schaefer in 1910 that this happened because of the lack of insulin. In 1921, Frederick Banting and Charles Best began extracting insulin from dogs' pancreases, and in 1922, Leonard Thompson gave this insulin to a boy, which saved his life. This was a life-changing discovery for many that suffered from the disease, and in fact, this discovery won a Nobel Prize in 1923. Eli Lilly, a large pharmaceutical company, began large-scale production of insulin by extracting it from cattle and pigs. It took 2 tons of animal tissue to produce 8 ounces, or 226.8 grams of insulin. There was a slight issue with this as it caused allergic reactions due to the insulin being from cattle and pigs instead of humans. Therefore, research began looking for new ways to create human insulin.
Obviously, the traditional process isn't exactly ideal, being expensive, messy, and inefficient. This resulted in the requirement for us to produce insulin in a more efficient way. Insulin, just like any other protein, is a chain of amino acids. Chemically synthesizing proteins is both complicated and expensive. Insulin is composed of 51 amino acids. It has a molecular mass of 5,808 daltons. Because of this, another novel way of converting amino acids into insulin would be required. Turns out the best thing for creating proteins, efficiently and cheaply, are living organisms, as every organism has to produce its own proteins to survive. Biocatalysis became the answer. This was done by integrating a stretch of coding human DNA into an E. coli host, resulting in an E. coli strain capable of producing human insulin. This insulin could then be collected and purified. Multiple changes were eventually made to the insulin DNA to optimize its synthesis in bacteria. In the future, Saccharomyces cerevisiae, or common yeast, may be a better host organism for human insulin, as it is able to more effectively complete structural modifications to the protein after its production, called post-transcriptional modifications, such as phosphorylation and acetylation, to assist the correct folding of the protein. Unfortunately, proteins produced by yeast possess several noticeable differences from naturally produced insulin, and are therefore easily detected by the immune system. This leads to the insulin having a short lifetime in the body. Research has been done to develop yeast-derived insulin that is less distinguishable from human insulin.
Colistin is another example of a drug that is biosynthesized. Colistin is a polymyxin and is used as an antibiotic for gram-negative bacteria. It is a large polypeptide that acts by interacting with the bacterial outer membrane and through displacement of certain ions results in cell death. Currently, it is used worldwide as it is one of the last antibiotics that gram-negative bacteria are not resistant to. Colistin, or polymyxin E, is produced from strains of Bacillus polymyxa through non-ribosomal peptide synthesis. Non-ribosomal peptide synthase is done by specific enzymes within the bacteria. The genes for this peptide are in the operon, or genomic DNA, of the bacteria. The enzymes are PMX-A or PMX-E and catalyze 4 or 5 adenylation reactions, respectively, to amino acids. The colistin can be obtained by incubation of the bacteria in a filtration process. This is done on an industrial scale and is the simplest way to get colistin, as it reduces the materials and time that would be needed if it was synthesized in the lab. Biosynthesis is a very useful way to produce drugs, as it eliminates a lot of materials needed in a normal reaction. It can also be difficult to create these long-chain organic compounds through synthetic routes, whereas enzymes can do them a lot quicker and easier than we can. They also produce higher yields and are generally more efficient than synthesis in the lab. Like our example with insulin, the previous methods of extracting it were difficult and wasteful, and to synthesize the insulin would have been even more difficult due to it being a series of complicated reactions. The same goes for colistin and any other large compounds. Therefore, it makes sense that biosynthesis would be a popular way to synthesize pharmaceuticals, as it saves manufacturers time, money, and resources.